Hi, everybody, and welcome to Neuroscientists Talk Shop, UT San Antonio's Neuroscience Research Podcast. Today is November 20th, 2025, and we're talking with Cristian Lasagna-Reeves. He's Associate Professor in the Department of Neurology at Baylor College of Medicine in Houston. Cristian studies proteins that are implicated in the mechanism of degeneration in neurodegenerative diseases, maybe lots of neurodegenerative diseases, how they are changed in the diseases and the changes that in the proteins that lead to the disease process, and his special interest in the tau protein, which seems to be ubiquitous, seems to be part of practically all the neurodegenerative diseases, so that makes it kind of unique and interesting. So, hi, Cristian. I hope I didn't get that too far off.
Oh, hi, how are you? Thank you so much for the invitation.
So also with us is Hyoung-gon Lee and George Perry, who are our local experts on mechanisms and processes in degenerative diseases. So I'm your host, Charlie Wilson. So Cristian, just for my benefit and for other listeners who are not up on the tau protein and neurodegenerative diseases, could you start by just giving us a little short thumbnail sketch of tau, its normal function, what goes wrong?
Yes, tau, as you said, is mainly expressing the brain, highly expressing neurons. The role we think now that has many other the roles, but the most the one that has been more studied or so is that has a role stabilizing microtubules. So it's highly enriching the axons of neurons, and it's highly dynamic. Attach, helping with the dynamic of microtubules within the neurons. And as you said, not only in Alzheimer's, but near 25 other neurodegenerative diseases, like frontotemporal dementia, progressive supranuclear palsy, even cases of Parkinson. Tau tends to, there is a hyperphosphorylation of this protein, tau, for reasons that we don't know, maybe activation of multiple kinases, phosphorylate tau. This inhibits the attachment of tau to microtubules, and there is an accumulation of tau in neurons mainly, and trigger the aggregation. And we see those accumulation aggregates of tau in neurons in the brain of patients, as I said, of Alzheimer's disease. And as you said, it's really interesting that in almost every single neurodegenerative diseases that we know, there is some form of tau aggregation as well.
So the first thing we would worry about is the hyperphosphorylation. That's the start of it?
Yes, like I would say that, and you guys can correct me if I'm wrong, we study the hyperphosphorylation because it was the phosphorylational modification or one of the hallmarks of these aggregates that we know we call tangles of tau. And it's what we see in post-mortem biopsies on patients, but still the debate is, I'm sure they are important, but how essential they are, this is a secondary effect of their aggregate. Maybe there are over 50 phosphorylation or 30 phosphorylations in tau. This is our physiological phosphorylation or happen only in disease. So we go back to the point that still there is so much that needs to be learned about the physiological role of tau and how tau behave in normal conditions and what goes wrong there that trigger this aggregation. So phosphorylation definitely should be an important event, but is the event that we want to tackle. There are clinical trials that have failed in the past that they try to inhibit these kinases in patients, and we don't see positive outcomes.
So we don't really know the sequence. It could be that phosphorylation happens after that.
Yeah, it could happen that, and which one is the one that could be important, or as you said, happen after the accumulation occur or the aggregation occur, really.
So the thing we know is that it is the polymerization of tau in the cytoplasm that creates these filaments that are bad.
Yeah, it's mainly in the axon and then accumulate in the cytosol, as you said. But again, we see when we analyze post-mortem brain samples from patients, we see the very final state, and we identify those aggregates, and we said, okay, maybe this is what was triggering, but what we were talking before is indeed that the toxic entity, or there is something else that we are not seeing at the very final state.
And usually there's some discussion of some misfolding event that happens to the monomer. Is that, how clear is that? I mean, what's it mean to say it's misfolded?
I think that there is a lot of in vitro data in cells, in mouse models that show that tau is a protein prone to misfolding to aggregation, and that initial misfolding trigger the multimerization and aggregation.
I see. So that misfolded tau isn't just going to hang around soluble, folding and misfolding and folding and misfolding.
All these proteins are really dynamic. So I think that you have all the chaperones that are trying to fold tau correctly. Then if it doesn't go well, you have the proteasome system, autophagy system that are going to get red things that are not working. So that's the reason I think that this is a really long process of this battle of correct folding in certain way. And that's the reason that we see these are diseases that happen with aging after many, many years, 70s, 80s, 90 years old patient. So, but during our entire life, there is this dynamic process of tau being folded correctly, then misfold, but then you have chaperones that make sure that it's folded correctly, the whole machinery, and if it's not, then you have another machinery to clean all this bad tau in certain way. But then with aging, you have oxidative stress, you have inflammation that can make that work a little bit more difficult for neurons, and then that equilibrium moves towards aggregation. And because another point is, all these aggregates that we see in the brain of patients after they die, whenever they do the autopsy, occurs many, many years before, even before patients have symptoms. So this is a really, really long process, that takes time and again, really, really dynamic.
So there's a bunch of these diseases and they all have it. Is the same tau in all of them? Or are there different taus in different diseases?
In different diseases, there are different tau. Tau has 6 isoforms. And how we can separate is there are isoforms, there are 3 of them, that they have 4 repeat domains that bind to microtubule. Remember, it's a microtubule associated protein, and the other 3 isoforms they have only 3 repeat domains that bind to microtubule. So we call the disease, the tauopathy, a 4R tauopathy or a 3R tauopathy, depending if are the isoforms with 4 repeats that aggregate or the isoform with 3 repeats. In the case of Alzheimer, it's 4R, 3R because all isoforms aggregate. And in a healthy brain, in certain way, you have equal amount of 4R and 3R tau. But then there is the equilibrium breaks, and there is an increase for some reason of 4R tau. And when there is that imbalance, also trigger the accumulation and aggregation of tau. So there is a lot of diversity within tauopathies, which isoform, which brain region, which type of cells you see that form or have these aggregates in postmortem samples. In ADs, only neurons. You have other tauopathies like PSP or CVD that you will see tau aggregates or phospho-tau accumulation in astrocyte or in oligodendrocyte as well. So that will depend on the disease.
And the place where this starts is where there's a lot of tau, I guess, where there's a lot of microtubules. So it starts in axons usually, I guess. That's where the most microtubules
Yeah, like 80-90% of tau in the brain is expressed on neurons. But that still doesn't answer the question of why we see aggregates in certain brain regions if you see tau in every single neuron.
Because there are microtubules everywhere.
Mm-hmm. Yeah, but why you don't see aggregates in the brainstem on patients with Alzheimer's if you have tau there as well?
Oh, yeah.
But you see it in PSP, but not in other areas, so in frontotemporal dementia, in frontal cortex, but you have tau, you should have tau everywhere in the brain, tau aggregates, but depending on the disease, you have only in specific regions. And that's a question that hasn't been answered, at least that I don't know the answer really. So
I think the important matter is that I think the cellular localization also kind of mislocalized. I think that's what you want to, also you covered that in your research and your seminar.
Mm-hmm.
That can be localization of the tau protein from the axon physiology, and then they kind of tend to go to the dendritic or presynaptic region when the pathological aggregations happen, right?
Yes.
So,
yeah. Yeah,
So I think this localization in the subcellular portion of the neuron is also an important pathological feature of the tauopathy, understanding the tauopathy, because that's been known that, there's such a mislocalization.
So I guess this partly raises the issue of how is tau bad? I mean, how do these aggregates harm the function of the cell?
I think that's what, yeah.
You're hitting right to the central question.
Yeah,
Right.
yeah, no, that's the big question is how which type of aggregates that we're seeing, if indeed it's bad, it's an effect of protective effect from the brain. They are again secondary to something. We cannot rule out. There is always all the evidence point to a gain of toxic function. These aggregates are toxic, but we cannot rule out that in certain aspects that there is a loss of function of tau as well, because it's aggregated, it's not binding to microtubules or helping with the dynamic. That is playing a role as well.
Oh, so it could destabilize microtubules, which would cause axonal transport to quit working well.
Yeah, like it has been shown that there is like in models that axonal transport impairment as well, when in the context of tau aggregation.
So when we say something like this is toxic, what we mean, I think, is we just added it to some cells and it killed them, right? But we don't know exactly how it killed them. We just know that it's
and we don't know we're applying it in a physiological way, right? When you're applying things to cells.
Yeah. It could be doing a lot of things at once that are...
Probably like most of biology.
Yeah. Yeah, that's the problem. When we say something like, we know this is toxic, it sounds like we know more than we know. All we know is it hurts the cell.
When we do it in that way.
Yeah.
Yeah.
Something else that's been observed, and what I'd like to know your thoughts, is that it's been described in development that many times tau develops phosphorylation patterns very similar to Alzheimer's disease.
Yeah.
And further, the tangles or accumulations of tau occur when people are quite young in certain regions of the brain, right in the 20s.
Yes.
What are your thoughts about those changes when there's not obviously pathology?
Oh, that's that's a difficult question, to be honest. But like regarding the phosphorylation is what we kind of discussed a little bit at the beginning. For instance, this new study that one of the newest biomarker for tau phosphorylation is phosphorylation at 217, amino acid 217, phosphotau 217. But there is a new study that showed that even in newborns, the level of phosphorylation of tau at 217 is really high and then goes down and then goes up again with 8.
And that type of finding, not necessarily that site, has been known for a long time, that during development, there's very similar changes in tau.
Yeah. Yeah, and there is a chain of isoform for what I mentioned.
There's a lot of cell death.
Tau.
There's a lot of cell death during development too, though.
Yeah. Yeah, no. That's the reason it's so important to really go back a little and understand the physiological role of tau. That we don't know well what is doing. These phosphorylations are only pathological, or they have a physiological role, maybe important during development, and then there is no phosphorylation during childhood and adulthood, but then comes again. So that, but there is, again, we really need to understand this protein more in detail before developing more treatments, again, targeting tau in the context of disease, So the isoforms, and it's something that in a technical point of view is important because the free R isoform, it's highly expressed during development. And then when you're born exactly there, you have equal amount of 4R with free R. And then in aging, you start getting more 4R. Right? But when you're working with iPSC cell lines or neurons, you have more free R, and that has been a challenge for the field. Why you have more free R during development, and that translates to the models that we have as well, the cell models, why you have free R and not 4R tau, the other isoform or the other group of isoforms. So definitely, maybe we're going here in circles or so, and not giving the exact answer, but tau has an important role during development, or has a role as well that we don't understand at all, really.
So maybe we're so interested in
the
how tau is bad, we don't think very much about what it normally does.
And then one of the studies that really kind of push that is a toxic gain of function that I believe on that is the fact that the tau knockout model is normal or roughly normal, but there is a compensation with other microtubule binding proteins. So there is a compensation mechanism that if you don't have tau, but the mouse express.
Well, that makes it hard to find out what tau is doing normally because you take it away and you don't see anything.
Yeah, and then you have other microtubule associated proteins that are upregulated, maybe compensating for that loss. So definitely we will be interested to develop other models, cellular or in vivo mouse models, that we can turn off tau and see what happens if you eliminate tau at different stages, or at least invest a little bit more in the physiological role, like the phosphorylations, the isoforms, why it goes up at early stages, phosphorylation that goes down and then up again, that there's a role there.
Do you think the phosphorylation controls the microtubules, or do you think it could be the other way around?
I think that certain phosphorylation, like in serine 56,
I know in vitro you can definitely phosphorylation control microtubules, but do you think in vivo that's what happens?
I think that can be that, but based on the in vitro and cellular model, but there are some phosphodies that kind of, if you phosphorylate certain sites, it's not going to bind. You dysphosphorylate it, it's going to bind. So I think that maybe that's a mechanism that how tau is binding to microtubule, that some phosphorylation block that binding. So if you don't want all the tau to be interacting with microtubule because there is not going to be microtubule dynamics. So how you control that attachment, detachment could be through phosphorylation.
I see. So that's what I was going to ask you, because from what you said, it sounded like people know what happens if you change the phosphorylation state of tau normally.
Specific to the microtubule.
In vitro and in cells.
Yeah.
But I did a study of looking at microtubule density, and it wasn't related to having a neurofibrillary tangle in the neuron at all.
You mean the human postmortem brain tissue, right?
No, biopsies.
Biopsy, yeah.
Biopsy tissue, there's about a 90% drop in microtubule density in Alzheimer's disease. But it has nothing to do with whether the neuron has a tangle.
But it could still have to do with phosphorylation of tau.
Could, because it doesn't have to be forming a tangle.
Yeah, so in vitro, when you're doing these experiments or in cells in the dish, you cause phosphorylation of tau. Is that, I mean, people say it stabilizes, tau stabilizes microtubules.
If it's in the non-phosphorylated form.
I see. So when you phosphorylate it, you can see microtubules destabilizing.
That's how it was discovered, sort of, the phosphorylation piece. First, it was discovered because tau was important in the in vitro assembly of microtubules. And then later on, it was discovered the phosphorylation. It was soon after, right? The phosphorylation was discovered still by Kirshner, I think.
I think, yeah.
And this is phosphorylation in general, so it doesn't relate to which sites are being phosphorylated or anything like that.
Yeah, I mean, people refine that. I don't know all the details of which site was important in doing it.
But there are a lot of phosphorylation sites upon the tau protein, and then some phosphorylation site is related the specific to the pathology, like for the Alzheimer's disease, and some phosphorylation site is kind of common in the development and then the pathology too. So I think that's a field still, I think we need looking further. I think nobody knows that clearly that what individual phosphorylation site, how they connected with the disease like that. So
the reason I'm sort of pursuing that is because I think I had picked up some kind of a very naive idea that tau polymerization was sort of like a crystal formation in a supersaturated solution, and there's just a lot of tau, and then like what happens in a supersaturated solution, you drop a crystal in and the whole thing just immediately crystallizes.
More like a liquid condensation.
So thinking about it kind of that way, you think, well, the cell just has a ton of tau, but it's all not misfolded. And then you throw in a little tiny bit of misfolded stuff in it that misfolds all the rest, and that's what goes on.
That's one idea.
Yeah.
But but it doesn't account for all of this complexity
No,
of
it doesn't.
different phosphorylation sites and different species.
And interacting proteins.
Interaction time. Other PTMs, acetylation has been shown to be important. The cleavage of tau, like there's evidence that needs to be cleaved N or C terminal to trigger the aggregation as
And
well.
also stabilizing the conformation chain. So oxidation plays some role in it also.
Right. So I think that's even more complicated because not only phosphorylation, like for example, acetylation and other kind of modifications happen to the tau protein, and they all demonstrate as linked with pathological development. So, it's very difficult to understand.
And they're linked together, like the oxidative modifications require phosphorylation.
So I think we should talk about other proteins that modify the tau process, because you've been studying those. And specifically, I was thinking of listening to you earlier that microtubules are everywhere in the axon, but it looked like the early stages of tau polymerization were happening at pre-synaptic terminals, which are not everywhere on the axon, and they're not the place where the microtubules are highly concentrated. They're the place where the microtubules are sort of dynamically falling apart.
Yeah, but you answered that question there. It's where they're dynamically folding apart. So you need tau in that moment to deattach, to allow them to be dynamically falling apart. So how, if we go with what we were just talking, there is that equilibrium phosphorylation, phosphorylation of tau exactly in that area, especially because you have microtubule stabilizing them, and then when you're going to get to the synapse, is where the microtubules are falling apart, you need the tau to deattach to allow that dynamic equilibrium. And that's where there is, I imagine there should be a phosphorylation mechanism and dephosphorylation. Because exactly there, like if you imagine the entire axon, the middle side, there is axonal transport, there is some dynamic, but the highly dynamic part of the microtubule is at the very beginning or the very end. And that's when the machinery has to be like running perfect. And that's when you go really fast, when things have to move with a lot of details, is where things start failing eventually. So that's why I believe that that's the key moment where tau can start this process that we have been talking.
But there are also some proteins in the presynaptic terminal that might contribute to that.
Yeah, like in my work, like we see many, and not only my group, like when several other groups, when they immunoprecipitate tau aggregate, it's highly enriched in synaptic proteins or mitochondrial proteins that we think that can be presynaptic mitochondria. And our research, we work in this protein basun, that is a presynaptic scaffolding protein, that indeed is and not only basun there, but you have piccolo, that are big proteins that are intrinsically disordered, that physiologically those proteins tend to form liquid phase, or they are prone to aggregation. So imagine now that we know more about these proteins, that when they're not working or not necessary for the cell machinery to use them, rather than to be degraded, they form this liquid phase, that it's another state of conformation. I think that occur, or based on the literature, occur a lot at the presynapse. So you have this equilibrium of changing foldings, tau coming from with the microtubule phosphorylating to allow the microtubule to disassemble. But then you have all these presynaptic proteins that they're moving vesicles, everything like 100 miles per hour, if you want to say in a certain way, and everything has to be working like a clock, like second after second, like perfect, and all these proteins that indeed are intrinsically disordered. They are moving depending, they're going to do a conformation depending with which other protein interact. There has to be a moment that things start not working really well, start failing. And for example, this protein that we are working with, basun, induced by mutation or some other stress, tend to accumulate and aggregate at the presynapse and create a really ideal environment for other protein prone to aggregation like tau to bind and then continue induce aggregation of tau as well. Or if tau start aggregating as well, but some can interact with this aggregate and make tau aggregates more stable, So there is this perfect equilibrium in the cell of proteins, and I think there is this study from this group, who is a review, like Fabrizio Chitti and Dobson, like many years ago, that they showed that many of these proteins, in an evolutionary point of view, they tend to aggregate. They are prone to misfolding. So, but we have a whole machinery, like mammals, we have a whole machinery of chaperones that avoid that misfolding and aggregation. So, imagine for many years your neurons working non-stop. Neurons do not divide. It's not like any other cell that, they stop working, they die, and then you generate another one, you lose your neuron, that's it, So, these proteins are pushing to aggregate, but the cells are avoiding that and continue working. Phosphorylation, dephosphorylation, microtubule dynamics, vesicles moving. So I think that create a moment that one start aggregating, inducing the aggregation of the next one. So
but then you're saying that this instability in proteins is a common feature. Why wasn't it selected out evolutionarily?
I think that we're still moving with evolution in certain
Thanks, George.
We just have to wait another billion years.
So, and I think it's a disease that we have to cure, but it's a disease that we are seeing in history. It's something relatively new. Like humans, we were not supposed to, or never, like in the last 50 years, we are increasing our age expectancy. So I think that that's something can be related. It's a disease that we have to work hard to find a cure, but in an evolutionary point of view, how many, I don't know, that will be interesting to see mammals that live much longer than us, if they develop certain type of protein misfolding or not. I'm not familiar with that. I know that bigger mammals, dogs, they develop also aggregation. Dolphins, they have tau aggregation as well. I don't know how long they live, so but it will be interesting to see that part as well.
Presynaptic terminal is the hardest working part of the brain, of the neuron. It's got all of the electrical stuff that's happening that somata and dendrites have, but then it has all the exocytosis and vesicle recycling and all of that stuff, and it's the starting point for retrograde transport. So it's like, and you can see they totally accumulate mitochondria because they have such a high, I suppose, because they have a high energy demand.
Yeah. So
because sometimes I've heard you say, a couple of times I've heard you say, maybe not right here yet, but I'm going to say that it's a kind of stress that triggers this, and I'm wondering, stress means different things. It means a force that could deform a piece of metal or something like that. But in this case, what do we mean when we think of something that stresses the biochemical mechanism in the cell?
I think can be different insults. For instance, now in the last more than a couple of years, one stress can be trauma brain injury. Now it's well accepted. With all the NFL players at the beginning, that the NFL tried to kind of avoid this type of research, but showed that in veterans, brain trauma trigger neurodegeneration, and it's called now CTE, chronic trauma encephalopathy, and you have a lot of tau aggregation in perivascular areas, depending where it could be the trauma. So in that case, that's the stress or the trauma. In Alzheimer, it's always, and you guys know much better than me all this, is you have two major hallmarks, that is amyloid and tau, and always has been which one is more important. Both are important, or none of them, So at the end, still the question is there, but my opinion can be maybe amyloid is not triggering the final toxicity directly, but amyloid, A beta plaques in Alzheimer have an important role to induce the hyperphosphorylation aggregation of tau. So in the case of Alzheimer, maybe the stressor for tau aggregation is the presence of amyloid. In other diseases like PSP, not always common, you have mutations in tau, not in every single case. So the mutation. So you need inflammation, like diabetes is really one of the strongest environmental risk factors for this type of diseases. So, and I don't want to miss anything, but there are many. So there is something that will trigger tau to start aggregating. Can be something genetic, can be a risk factor like ApoE4, can be amyloid, can be diet. Has been shown again, like following like Mediterranean diet could be a little bit more protective than other type of diet. High blood pressure also is an important factor. So that producing certain inflammatory response or oxidative stress in general could be the first step, like a mild response to trigger disaggregation of tau that then will loop and enhance those other events, So there has to be something that start, like trigger all these events, really. And not in every single disease, it's going to be the same.
Yeah, that's sort of not the answer I was dreaming of, because I would like there to be a kind of final common pathway of all these things, like the cell doesn't have enough energy, and in the end, it just didn't have enough energy, or it doesn't have enough some other important resource.
Hopefully, will be that, and we have one treatment for every single disease.
Yeah, that's the reason I think.
But I think not everybody, like maybe that was the idea, but each time now with the cryo-EM, they're showing that the structure of tau is different depending the disease. That will be relevant for a treatment, I don't know, but also we cannot be. I would love to be naive and thinking, we one antibody for immunotherapy, but the conformation is different, so the epitope is different. The antibody will recognize for an immunotherapy against tau, or a kinase to phosphorylate, but the pattern of phosphorylation of tau can be different depending the disease. So there are pros and cons. It's like hopefully, but doesn't look like that one treatment will be beneficial for all of them, because in Alzheimer's still you have amyloid and you have you have other factors. But also, if something that failed for Alzheimer's treatment, doesn't mean that it's not going to work for other diseases. So not to give up on those approaches as well.
So the simplest idea is you have too much tau, we just decrease how much tau is in your brain. Is that feasible to do?
Yes.
Because you said that a tau knockout has no phenotype.
So that was the base, and now I finished in I think in March they're going to release the data, phase 3, I think, for the ASOs, again, MAPT, that is the gene for tau. Phase 1 looks promising. I think CSF, there is a decrease on phospho-tau. But we don't know the results on the clinical trials. Now, if those ASOs, antisense oligonucleotides, the idea will be to downregulate, because that's what you said. What I was told many years ago by a lot of really important scientists, that is, hey, why you worry if there are the aggregates here or the oligomers? It's like, just get rid of tau, and you will, and then you avoid the problem. But there are two questions, and the results for 2026 on those clinical trials will answer that question. One is, will be too late after you have symptoms? You really need to identify the patient, and that's important of biomarkers way before the symptom appear, because if you have somebody already with major memory impairment, how beneficial will be to downregulate the expression of tau or the level of tau if the damage is already there. That's one thing. And the other one, going back to what we have been talking, is understanding the physiological role of tau, because we know that the knockout is normal, but decreasing tau in an adult, we don't know if will be beneficial if tau is doing something important. So we'll know next year.
There's some reason to think it would be safe, but we don't know whether it will be effective or not, right? That's what you just said.
Yeah, because
And-
we'll pass already the task, it will be effective or not. Hopefully, yeah. But
Are you guys all in on just knocking down tau?
No,
no,
why
I think tau is still these are all parts of responses that are happening during the disease, and we don't know how they really fit in.
So what's your
For one thing,
prediction?
there's a strong antioxidant activity from tau accumulation. There's an enzyme that's co-induced with it called heme oxygenase. We think it probably plays a role in turning over mitochondrial proteins. So I don't think it's just doing a passive random toxicity thing. I think it's part of some program that we don't understand when cells are insulted.
So what's your suggestion?
Well, I think if I was dealing with something, I had to make a guess, it'd be both energetics and inflammation. And I think those are upstream events, or either concurrent or upstream events. And tau plays a critical role, same as amyloid plays a critical role. And certainly intervening with it, it might be you'd want to decrease its thing. But I wouldn't say blanketly I'd want to get rid of it. It might make things worse.
Yeah.
That's what's happened with, I think it's the case that the trials that people tried addressing tau in less sophisticated ways so far have been therapeutically not of value.
Oh,
correct?
yeah, no,
In fact, the patients got worse.
Yeah, many trials targeting tau already failed. Other approaches inhibit aggregation, phosphorylation. They have
Good
been
patients
immunotherapy.
have not been a benefit,
Yeah.
and the amyloid beta has been the same.
Yeah.
The benefit is totally unclear. In fact, it may have had no benefit.
Okay, I'm trying to certify that both party here.
Yeah.
Like, and we briefly discussed with him, and then after seminar that I think we talk about different species of the tau aggregate. Like, we talk about you also kind of agree on me that some tau aggregate may be just protective, like, the later stage of the formation, like, limited that the spreading out toxicity, and then some of them, like, maybe I'm not sure you would gonna go to like a soluble tau or like oligomer tau would be the toxic species targeting like that. So don't you think that maybe kind of current approach? I know that some of them kind of specifically kind of targeting like oligo tau specific antibody. I'm not sure what's the current stage at the moment, but would it be kind of find some some foundation to kind of make happiness on the side, like that way. Kind of
We
compromise.
are saying
kind of,
something so different.
Yeah, no, nothing similar. I'm hopeful that will work because I'm trying to be positive.
Yeah,
but,
it could be. It's a
I don't want
possibility.
to say it's not going to work, but yeah, there is a lot of chances that because you don't want to get rid of all the tau or you don't know if it's good. And most important, even if you get rid of 100% of tau and there is no detrimental effect, we don't know if indeed is what triggered all the events or the key. So we know through animal models or cells some evidence.
Or
but
if it's a protective response part of that thing.
It's a protective effect. So no, I completely agree. Like when I said I hope that works, is because
it would be
obviously
great.
we all hope that we will find a cure, but obviously there's a big chance that maybe will not work.
I hope it works, and it's not pharma companies just manipulating statistics, which is what we've seen for amyloid so far.
Yeah, certainly with and that's the other thing. Maybe not moving a little bit from tau with all this treatment. They said the anti-amyloid treatment is not working ideally or really well or not at all according to some people. The benefits are very modest. So now what they are thinking is why we do a combinatory treatment, anti-amyloid and anti-tau. And my concern with that, or anti-inflammation, but my concern with that is for the anti-amyloid, it's not that, I'm being diagnosed with Alzheimer's, I can get the immunotherapy. It has to be an early state. It's not for every single patient. Your ApoE plays a role. Your ApoE genotype plays a role. So let's say I'm throwing a number here, but only 20% of patients with Alzheimer's can receive this anti-amyloid treatment. If we do we combine it with an anti-tau, from those 20%, maybe a quarter. So we're talking about maybe 5% of the total population is going to be able to get a treatment. So will be ideal to get something first economically affordable for everybody. I know it's difficult, that part, but also hopefully we can find something that it can be beneficial for a broader range of patients, So that's.
Well, in addition to that, I think
Yeah.
I think that's one of the most significance of your finding, your discovery in your research. You basically demonstrate that specific population of the tau, using the fractionation, you demonstrate that only a relatively small portion of the tau you found it is actually working with your bassoon protein and the aggregation and spreading type of a thing, and then also you show that functional study. So I think your study actually suggesting that we better go to target the specific population of the tau aggregate or seed, whatever we call it, rather than just bluntly target all tau.
Yeah, no, that's
I-
kind of
Another question: when you have, it's called toxicity, as Dr. Wilson pointed out. Basically, you're talking about molecules that have a lot of activity, biological activity. We denote that as toxicity, but it could just as easily be activity as biological, and you're at a different concentration range, you're out of context.
Yeah. So it seems to me as an outsider that the method of discovery built around clinical trials is extremely inefficient. That every time we come up with we have a popular idea, we come up with a treatment based on our idea, and then we do a multi-million-dollar clinical trial, billion-dollar clinical trial, and the results are not what we wanted. And we end up making excuses. Oh, well, it would have worked if we had done it this and we've done it that. We need to do the whole thing over again now with this modification. Well, in animal experiments, we do that, or with cells in a dish. Oh, I think I left out a reagent. Tomorrow I'll take some cells.
And you have your result within a few days.
Yeah, but if I'm going to spend millions and take 10 years, then that's a very slow way for us to refine our idea, I think. So I don't know.
I think that's a challenge. Everybody can recognize it. Yeah. So welcome to the world.
Okay, well, thanks a lot. I think we should end there. So thanks, Cristian.
No, thank you so much for the invitation. Really appreciate it.
George?
Thank you.
Hyoung-gon?
Yeah, thank you. I think it was a very nice discussion.
This has been Neuroscientists Talk Shop. Okay.